everyone. I am Rajiv Saxena, Associate Professor, Smriti College of Pharmaceutical Education, Indore. In today's video lecture, we'll discuss about the phase contrast microscopy. The phase contrast microscopy is having a very wider application and it is having so many advantages as compared to that of the bright field or any other class of the microscopy. Suppose if we want to visualize an cell or an object maybe in the living form, then it is actually not possible with the help of the bright field microscopy. Because in case of bright field microscopy, what we have to do? We have to go for the staining of the cell before the visualization. But because of the staining, the toxicity that will be generated by the stain, then it may happen that the cell that we are staining is actually killed by the dye. So in that case we are not able to see the cell under the live condition. So if we want to see the cell under the living condition, in that case the phase contrast microscopy plays a very important role. Suppose we want to go for the analysis of the growth of the cell, we want to demonstrate the growth of the cell, then also we can go for the use of the phase contrast microscopy, because in this case we don't have to go for the staining procedure that may be toxic to the cell. Now coming to the basic layout of the phase contrast microscopy. As you can see in this diagram, this is the basic arrangement that initially it will have the light source, then it is having the presence of the annular ring, then we have the presence of the condenser lens, then it is having the part where we can keep the specimen, that is the specimen holder cell, then we have the objective lens, and at the end we have the face plate. So this is the basic layout or the arrangement of the phase contrast microscopy. As you can see that in case of microscopy, so many lenses, they are actually very common. Like you must have heard about the condenser lens, objective lens, eyepiece lens. These are the lenses which are very common in case of all the different kind of the microscopy. But especially if you are talking about the phase contrast microscopy, as you can see in this diagram, two special features are actually available. Number one is the presence of the annular ring, and number two is the presence of the face plate. These type of arrangements are actually not available with any other class of the microscopy, and these arrangements basically makes this microscopy special, by which we can go for the visualization of the cell under the living conditions. So one by one we'll understand how these different parts of this phase contrast microscopy plays an important role in the visualization of an object. Now, the microscopy starts with the light source. Like every microscopy, there is a certain sources with which we can go for the visualization of the object. Like in case of, if we are talking about the light microscope, then light is actually the source. Suppose if we are talking about the electron microscopy, then electrons are actually the source. So similarly, if we are talking about the phase contrast microscopy, then some light sources are actually available. It means here we can see the object with the aid of the lights. Okay, so that's why initially it is having the source of the light that will generate the light in the microscopy. But once the light will be generated, it will be focused towards the annular ring. Now the annular ring, as you can see in this diagram, it is having some structural components, and some solid structures are actually available in this annular ring that will interfere with the passage of this light source. Okay, as you can see in this structure, some hollow spaces are actually available that forms a spherical ring space. So in that ring, some space is available that can only permit the light to pass through it. Otherwise, whatever the solid structure or the solid bodies are available in the annular ring, they will not permit the passage of the light through it. Okay, so what will happen? The light, when it passes through the annular ring, it will be available in the form of the hollow cylinder. It means the light is not available in a continuous fashion, but rather it will be available in the form of some hollow cylinder. So in that form, the light will be focused towards the condenser lens, and from the condenser lens it will focus towards the specimen. Specimen can be your any cell, maybe for example bacterial cell that we want to visualize under our microscopy. Okay, so what are those arrangement? Initially we have the light source that will go for the generation of the light, then we'll have the annular ring that will permits only the light in a particular fashion, maybe in the form of the hollow cylinder, and it will retard or it will not permit the entire light that will be generated from the light source towards the condenser lens, but rather the light will be focused on the condenser lens in the form of some hollow cylinders. Then from the condenser lens, the light will be focused towards the specimen. Now, when the light will be focused towards the specimen, we know that the light actually travels in the form of the wave, and every wave or every light is having certain wavelength. And what is the definition of the wavelength? Wavelength is actually the distance between the crest of two adjacent waves. As you can see in this diagram, this is first crest, this is second crest. So the two crests, the distance, if we calculate the distance which is available between the two crests of the adjacent wave, then it will be called as the wavelength. So whatever the light source that we are using, accordingly it will generate the wavelength, and that wavelength will be focused towards the specimen that we have kept in the specimen section. So light, when it is generated from the light source, it will enter into the annular ring, then it will enter into the condenser lens, and from the condenser lens it will be focused towards the specimen. Now this light is actually focused towards the specimen, but when it will enter the specimen, say for example it is having some cell structure. Okay, so as you can see in this diagram, this cell is having the presence of the nucleus. Okay, nucleus is available. So if when the light will enter towards this solid structure, so this solid structure, we can say that in a scientific terminology, we can say that it will have some high optical density, or it will provide some high refractive index. So because of that high density or because of that refractive index, the light that will come out of the object will have somewhat different wavelength from the light that we have entered inside the specimen. As you can see in this diagram also, like when the light is actually entering inside the cell or inside the specimen, it is having some different wavelength, whereas the light, because of the optical density, because of the enhanced optical density, because of the refractive index, the light that is coming out of the specimen, it will have some different wavelength. Okay, so what will happen? As you can see in this diagram, there are two different kinds of rays are actually visible. Number one is a retarded light rays, and number two is a unretarded light rays. Now what is the difference between the retarded light rays and unretarded light rays? Suppose if the light, it is passing maybe away from the object, so these waves, they will not be interfered by any part of the specimen. So these waves are actually called as the unretarded light rays, whereas the rays or the wave that will enter inside the specimen, they may be retarded. Maybe there is some change in the wavelength because of the presence of the solid structure, maybe because of the presence of certain organelles. So these radiations will be called as the retarded rays. Okay, so what happens? These light, it will be generated from the light source, it will enter into the annular ring, then it will enter into the condenser lens, and from the condenser lens it will be entering into the specimen cell. Now in this specimen we have already kept the sample, so whatever the light that is actually entering in the specimen, then what will happen? There is some change, there is some retardation of the light will takes place, and here we can differentiate the light rays into two categories. Number one is a retarded light rays, and number two is a unretarded light rays. What are unretarded light rays? Those light rays which are not being interfered by any solid structure or any bodies which is available in this specimen. Whereas what are the retarded light rays? So these are the light radiations that will be retarded because of the presence of some organelle structure or organelles inside the cell or the specimen. So whatever the lights that will come out of the specimen, it is having two categories. Number one, retarded light rays, and number two is a unretarded light rays. Then with the help of this objective lens, it will be so focused towards the phase ring. Now the phase ring is also having some special function in case of the phase contrast microscopy. As we have seen in case of the annular ring is responsible for focusing the light in the form of some hollow cylinder, whereas the phase plate is also having some special feature. Now we know that we are concluding that when the light will passes through the specimen, then it will be differentiated into some unretarded or retarded light rays, and we are also calling it like that there is some change in the wavelength before they entering to the specimen and after coming out of the specimen. But this change in the wavelengths, they cannot be seen by our eyes. Means we cannot see that there is some change in the wavelength. Okay, but what we can see the change in the intensities of the light. Okay, these terms are very important. Like whatever the light that is entering into the specimen, it is entering in the form of the wavelength. Okay, and here what we are saying that when it will come out of the specimen, it will have some different wavelength. But it means there is some change in the wavelength, but that change cannot be visualized by our eye. Okay, so what we are doing, we are using here the phase plate. So what phase plate is doing, we know that we cannot assume or we cannot see the change in the wavelength, but we can easily see the change in the intensity of the light. We know that we can easily differentiate between a very high light or a very dim light or some medium intensity light, so that intensities can easily be visualized by our eyes. So what here the phase plate is doing, that phase plate is responsible for, as you can see that these are the unretarded light. Okay, so unretarded light, the phase plate will, what the phase plate will do, it will go for the constructive interference of the unretarded light rays. As you can see, like this is the first wavelength and this is the second wavelength. So what phase plate will do, it will combine these two waves and produces a very high intensity light. So it will have a high intensity that can be seen in the eyepiece or at the site where we are actually visualizing the object. But what it is doing also, whatever the radiations that we are calling it as a retarded lights, so these retarded lights will undergoes the mechanism of destructive interference with the help of the phase plate. And because of that destructive interference, we can go for the visualization of the light in the form of some low intensity as compared to that of the constructive interference light generated that will have the high intensity. So here actually there is some differentiation in the intensity of the light, that because of the constructive interference some high intensity lights will be generated, and in between that high intensity light there is a presence of some low intensity light, and as a result of which a contrast will be created. That is actually the basis of the phase contrast microscopy. It means here we are trying to generate some contrast between the intensities of the light, some high intensity light and some low intensity light. So if some low intensities are available inside the high intensity light, then definitely we are able to see the object in a very magnified way and without the staining procedure. Now the phase ring is also responsible for enhancing the unretarded light rays by lambda by 4, whereas reducing the retarded lights by lambda by 4. It means here the phase plate is also performing an important function, that it will try to differentiate the intensities of the light. It will try to differentiate the high intensity light and the low intensity light. As a result of which a very clear contrast will be generated, and because of that contrast, whatever the cell, whatever the organelle is that we need to visualize, that can be easily seen with the help of the phase contrast microscopy. So summarizing here, phase contrast microscopy is a very essential tool in the field of microbiology, in which we are able to see the cells under the live condition. This is the basic arrangement of the phase contrast microscopy. It is having the light source, then it is having the arrangement of the annular ring. The light will process from the light source towards the annular ring, then it will be focused towards the condenser lens, then from the condenser lens it will focus towards the specimen, from the specimen it will come out and it will be focused towards the objective lens, and from the objective lens it will enter into the phase plate, and after the phase plate it will, then we can see the object with the help of the eyepiece, and we can easily get the contrast, and because of that contrast the objects are clearly visible. Thank you.